So now that we've established some information models for the cortex, let's use these models to describe changes to conditions and receptive fields consistently on multiple levels of detail. In other words, a hierarchy of description. First, I'll describe the process at high level in terms of cortical areas and columns. Then, map a key part of that description to the internal cortical column level. Then, a part of the column level description to receptive field expansion at the pyramidal neuron level. Then, to the dendritic branch level. Then, mapping several parts of the branch level description to descriptions in terms of chemistry within the synapse and the neuron. The key point is to demonstrate how using the recommendation and condition definition detection paradigms makes this type of multi-level description possible. So, starting at the level of columns within the cortex. Suppose this is a conceptual view of the columns within one area, and this area detects receptive fields that are able to discriminate between visual perceptions of different types of animal. One individual column won't correspond with a type of animal, in the sense that the receptive field would be detected in every perception of that type of animal and not in any other type. For example, as we discussed earlier, if the columns in red were those that sometimes detected their receptive fields in bird experiences, only a subset would be detected in any one experience, and all the columns would also detect their receptive fields in some experiences of other types of animal. Discrimination means that the various subsets of columns that detect their receptive fields in response to different bird experiences are sufficiently similar to each other and sufficiently different from the groups of receptive fields detected in response to, say, bats, sufficiently different even though there's often overlap because of some visual similarities, that it's possible to assign recommendation weights in the basal ganglia to the receptive fields such that the strongest recommendation is almost always the appropriate one. So there have to be enough receptive fields detected in any situation to make that kind of discrimination possible. Now, the focus here is on receptive field expansions. So, as before, let's add another drawing of the same area, and suppose the requirement is that at least 6 columns detect their receptive fields in response to any input to the area. Because there's been extensive past learning, usually the columns that detect their receptive fields in response to a familiar bird will meet the minimum for any normally bird experience. But as in the example we discussed before, suppose you haven't seen a penguin before. So because of the novelty, less than the minimum number of receptive fields will be detected. Some will have bird-appropriate recommendation strengths and some won't. Receptive field expansions occur in columns with some similarities to the current circumstances. Again, some expansions to columns that have existing bird-appropriate recommendation strengths, some that don't. Although all these columns will in due course get some recommendation strengths in favor of identifying a bird. But for the moment, what I want to take to the next level is how the best columns for expansion are identified. At the column level, there are a number of different layers of pyramidal neurons, which I've simplified to 3. With the layers illustrated in order of connectivity, rather than the physical order, there's layer 4 at the top, layer 2, 3 in the middle, and layer 5, 6 at the bottom. So inputs from outside the column arrive at layer 4, outputs from layer 4 go to layers 2, 3, outputs from layers 2, 3 go to layers 5, 6, and the outputs from layers 5, 6 are the receptive field detections that recommend behaviors. This pattern of connectivity means that the receptive fields detected by neurons in layer 4 are relatively simple, the receptive fields detected in layers 2, 3 are a bit more complex, and the fields detected in layers 5, 6 are even more complex, which will mean that sometimes a few fields might be detected in layer 4, but nothing in layers 2, 3, 5 and 6. And sometimes there could be a few fields detected in layers 2, 3, but nothing in layers 5 and 6. Receptive field detections in layers 5 and 6 mean the column is detecting its receptive field, so no receptive field expansions are appropriate. But in this situation, with no output but significant receptive field detection in the middle layers, the column could be a good candidate for expansion if needed, because the strong activity in the middle layers suggests that only a small change would be sufficient to generate a detection. So signals indicating strong internal activity will go to the change manager, and if this column is one of the most appropriate, signals will come back to drive expansions. So let's have a look at a piece of this process at the neuron level. Ultimately, column receptive field changes have to be implemented by neuron changes, especially neurons in the output layers 5 and 6. Thinking of the receptive field of a neuron as a pot of conditions, the receptive field can be added by adding conditions to the pot. Conditions are instantiated by terminal branches on the dendritic tree, and the field could be expanded, for example, by adding a branch corresponding with, say, the condition C5. Let's go to an even more detailed level of description, remembering that we need to implement the information process of adding conditions to the receptive field. The existing conditions, like C1, have a number of fairly strong synapses, and there could be a number of subsets of these synapses which, if they all received incoming action potentials at the same time, could encourage the neuron to fire, in other words, a receptive field detection. So actually, the branch labelled C1 and the other existing branches detect sets of individual conditions, all very similar to each other because they share many of their inputs. But now think about the new branch labelled C5. The synaptic weights of inputs to this branch from within the column are all very small, but it also has an input from the change manager, which is large. How does this help? Well, consider that branch. The size of the synapses indicate their weight. The weights of the synapses that would define the branch condition are very small, although the synapse from the change manager is much stronger. This means that even if a large proportion of the condition-defining synapses are active at the same time, there won't be enough total postsynaptic potential injected by those synapses to initiate a calcium action potential deeper into the dendrite that would contribute to the neuron detecting its receptive field. In other words, no conditioner conditions are actually defined by this branch. But now suppose that the same inputs are active, but there's also an input from the change manager, indicating that the column in which this neuron is located has been selected for receptive field expansion. With this additional input, there's sufficient postsynaptic potential for the branch to initiate a calcium action potential towards the soma, which, if combined with other branch inputs, could cause the neuron to fire. If it does fire, indicating receptive field detection, it also initiates a back-propagating action potential into its dendritic tree. This back-propagating action potential increases the weights of any recently active cortical input synapses. These long-term synaptic weight increases to the condition-defining inputs on the branch mean that a similar set of inputs in the future will cause the branch to contribute to causing the neuron to fire, independent of whether or not there's an input from the change manager. So, in information terms, a condition has been added to the receptive field of the neuron. Note that this is one example of a change, and it involves a previously ineffective branch. All the cortical synapses could even be silent with zero starting weight. But a branch that's already detecting conditions could also add it to its portfolio with the aid of a change manager input, by initiating an initial silent synapse or just by increasing the weights of its other synapses. Notice that, as expected, you only add a condition if the neuron fires. Conditions are only added if quite a lot of already programmed conditions are already being detected. We've talked about new dendritic branches with zero-weight synapses apart from the change manager input, and new zero-weight synapses added to existing branches. One important question we touched on in an earlier lecture is how can these inputs be identified in a way that will limit creation of useless synapses? And as we'll discuss later, identifying appropriate inputs is an important role of sleep. Now let's describe a piece of this process on an even deeper layer. The synaptic change mechanism, based on temporal correlation between synaptic inputs and neuron firing, is widespread in the cortex. If a back-propagating action potential arrives at the synapse shortly after an input action potential, the weight of the synapse increases. And it's also observed that if the input action potential arrives after the neurons fired, the synaptic weight decreases. But note that on average, increases are larger than decreases. In information terms, the primary mechanism is receptive field expansion. But on the other hand, if a synaptic input frequently arrives after the neurons detected its receptive field, the implication is that the synapse isn't really contributing to the neuron receptive field detection, and its influence can be safely decreased. But this curve means that one contribution to receptive field detection outweighs several irrelevant occasions. So let's have a look at some of the chemistry that supports the higher level mechanisms. And this is an illustration of a cortical pyramidal synapse taken from earlier in the course. And in this example, the synapse is silent. The postsynaptic neuron membrane doesn't have any AMPA sodium channels, so there won't be any sodium current when glutamate neurotransmitter is released in response to an incoming action potential. But the postsynaptic membrane does have NMDA receptors. These NMDA receptors are ion channels that, when open, pass mainly calcium ions. Although they open in response to glutamate, the open channels, even when they're open, are generally blocked by a magnesium ion. When an action potential arrives from the presynaptic neuron, the presynaptic calcium channels open, triggering release of glutamate into the synapse. Glutamate binds to some of the NMDA receptors, but they're blocked by the magnesium. If the NMDA is still bound to glutamate when a back-propagating action potential arrives from the soma, the magnesium block is removed. The NMDA channels pass calcium, and the calcium ions initiate a cascade of chemical reactions that results in addition of AMPA channels to the synapse. So the silent synapse has now acquired some synaptic weight and can contribute to firing of its branch in the future. Going even more deeply at the mechanism, glutamate synapses onto a pyramidal neuron dendrite form at the end of spines. The size and shape of the spine is maintained by polymerization of actin molecules. The AMPA molecules that give the synapse its weight are located in the postsynaptic density, which is the region in the postsynaptic neuron that faces the synaptic cleft. Close to the base of the spine are ribosomes containing the messenger RNA that codes for AMPA molecules, and if activated, they'll synthesize more AMPA. Part of the chemical mechanism for increasing synaptic weight is that entering calcium ions trigger a cascade of chemical reactions that lead to activation of the mRNA coding for AMPA proteins. More AMPA molecules are synthesized, inserted into the neuron membrane, and diffused to the postsynaptic density, increasing the synaptic weight. However, there's a tendency for AMPA molecules to circulate between the postsynaptic density and the membrane, maintaining an equilibrium concentration in the postsynaptic density. If the size of the synapse doesn't change, the increase in AMPA decays away over a period of about an hour. However, there can be a dopamine synapse located close to the spine. If the dopamine synapse receives an action potential while the AMPA density is still high, it sends a signal to the nucleus of the neuron where the DNA is located. This signal encourages the synthesis of messenger RNA that codes for actin, transport of that mRNA to the region of the spine, and local synthesis of actin. The actin polymerizes in the spine, driving an increase in its size and extending the duration of the synaptic weight change. And we can look even more closely at the chemical processes. There's actually a lot of different receptors for different neurotransmitters in the neural membrane. I've illustrated one receptor for dopamine, the D1 type, which is a G protein receptor, and two receptors for glutamate. One is the NMDA receptor that opens only in response to a combination of glutamate and voltage. The other is a G protein receptor called mGluR5, and one important way in which G protein receptors act is via other molecules embedded in the membrane, like adenylyl cyclase or kinases like PKC. But remember, there are many more neurotransmitters and many more receptors that can act on a neuron that are not illustrated. And many of these neurotransmitters and receptors can influence the activation of genetic information in the neuron nucleus. And such activations occur when molecules targeting specific strands of DNA are themselves activated. A critical point to note is that activation of just one molecule can occur via multiple routes. For example, the activator molecule CREB can be activated by dopamine via the D1 receptor and the pKa kinase. The same activator is also targeted by the influx of calcium ions through an NMDA receptor, again via kinases. The G protein glutamate receptor can also target CREB via a kinase cascade. And the same receptor has another route which releases calcium ions from internal stores where they're bound in an inactive form. So there are multiple routes by which an external neurotransmitter can influence activity of the same genetic information, such as the synthesis of the mRNA coding for the skeleton protein actin. In addition, there's crosstalk between these parallel paths. Activity in one path can encourage activity in another path or inhibit activity in another path. So there's a very complex network of chemical processes linking external influences to structural changes in the neuron. So, how can we understand this network in information terms? Well, a neurochemical process implements an information process. And once again, such processes can be seen as condition detections that recommend behaviors. The presence of a neurotransmitter in the neighborhood of the neuron is a condition, and the receptors detect those conditions and recommend behaviors. These behaviors are often the initiations of cascades of kinase reactions, and the presence of such cascades are themselves condition detections that recommend behaviors. The recommended behaviors are different structural changes to the neuron. A structural change, like a long-term increase in the weight of a specific synapse, is a behavior which will occur only if it's recommended by a large enough combination of chemical conditions. It's worth noting at this point that one reason for the persistence of these information models, even at this detailed chemical level, is the need for evolution to occur at all. Because evolution depends on the occurrence of random mutations, which in some cases confer survival benefit, and these random mutations will be to chemical processes. If every chemical condition was a command, any mutation would have a high probability of changing or blocking a critical chemical process and would probably be fatal to the organism. So mutations wouldn't be possible. But because all the chemical conditions are only recommendations, the initiation of any one chemical process will generally be supported by a combination of chemical condition detections that all recommend the process. So if a mutation occurs that has some behavioral benefit in one area, it may also remove one of the recommendations for a chemical process critical in another area, but other recommendations will still be in place. So there's a chance that the undesirable side effects of the mutation won't be too severe. So the use of recommendation information paradigms at the chemical level is essential if evolution is going to occur at all. In other words, evolution itself is also a form of learning from experience by a complex system and subject to many of the same constraints. So what we've done is establish a description of the receptive field expansion process at the cortical area level, and then map critical small parts of that description through columns, neurons, dendritic branches, synapses, and down to neurochemistry. And later in the course, we'll discuss examples of similar hierarchies of description for complete cognitive tasks. Although a key point is that once we know that we can map high-level descriptions down to detailed levels, that tells us that we can just use the higher-level descriptions most of the time to understand cognitive processes, because we've already established the existence of the deeper-level processes that implement the higher levels, which is a bit like we can design features of an electronic system, and most of the time we don't have to worry about whether our feature can be implemented by integrated circuits and transistors, because the existence of the deeper level processes that implement the higher levels, in this case, are well known.